For most of the 20th century, science treated the adult mammalian brain as a static, finished product, a myth that is now shattered. Once childhood development ended, the neural circuitry was considered hardwired, entering a terminal phase of gradual decline. We now know that framework is entirely backwards. The brain is a perpetually updating system, continuously restructuring itself in response to environmental stimuli, learning, and physical trauma. This adaptation occurs through two distinct modalities. Structural plasticity physically alters the anatomical tissue, while functional plasticity reallocates cognitive logic and processing tasks to different areas of the network. To understand how the adult brain rewrites its own architecture without collapsing, we have to trace the precise biochemical triggers that initiate these changes and the strict homeostatic constraints that keep them in check. The adult brain operates as a fluid architecture, capable of deliberate evolution throughout an individual's lifespan. The foundation of this adaptable architecture begins at the microscopic level with activity-dependent synaptic plasticity. It follows a principle known as the Hebbian rule. Neurons that fire together wire together. When pre- and postsynaptic neurons activate repeatedly and in sync, the connection between them becomes stronger. This process is called long-term potentiation, or LTP. LTP requires a specific chemical sequence. Electrical signals cause the presynaptic neuron to release glutamate, activating NMDA receptors. These open, allowing calcium ions to flood the cell, triggering the embedding of additional AMPA receptors to permanently thicken the connection. These localized microscopic chemical events are what create the biological foundation necessary for all macroscopic learning and memory storage. The chemical cascade of LTP directly drives structural plasticity, resulting in the physical remodeling of the brain's cellular tissue. Neurons physically sprout new micron-sized protrusions along their branches, called dendritic spines, to solidify these new neural pathways. In certain areas, like the dentate gyrus of the hippocampus, the adult brain even undergoes neurogenesis, generating entirely new neurons to process incoming information. This physical growth and integration of new cells is heavily mediated by Brain-Derived Neurotrophic Factor, or BDNF, which acts as the primary molecular fertilizer for the nervous system. A brain that only adds new connections faces a geometric and metabolic problem. Without a way to remove unused pathways, the network would quickly become overcrowded, inefficient, and prone to failure. To counter this, the brain employs regressive plasticity to optimize the network. First, synaptic pruning dissolves unused connections. Second, apoptosis ensures programmed death of entirely non-functional neurons. Finally, axon retraction withdraws specific local branches to refine connectivity without killing the cell. The ability to learn and adapt relies just as heavily on this deliberate destruction and optimization as it does on growth. Zooming out to the macroscopic level of the cerebral cortex reveals the brain's second major adaptive mechanism, functional plasticity. This is the capacity to shift the execution of specific tasks away from damaged or underutilized tissue, reallocating that logic to healthy areas. While structural plasticity takes days or months to physically build new networks, functional plasticity operates rapidly to preserve operational capacity and ensure immediate survival. We see this clearly in clinical rehabilitation following a focal stroke or a traumatic brain injury, where eloquent cortex tissue controlling specific motor functions is permanently destroyed. The brain initiates homologous area adaptation, rerouting network signals so the exact opposite hemisphere adopts the functions of the damaged region. Similarly, in cross-modal reassignment during sensory deprivation, adjacent healthy regions expand to hijack idle tissue to process new signals. This reallocation relates to a phenomenon called diaskesis, where severe functional, metabolic, and electrophysiological alterations ripple into healthy tissue located far outside the actual zone of focal damage. While diaskesis causes initial impairment across the network, the reversal of these distant alterations is actually the primary mechanism that drives spontaneous functional recovery in the months following an injury. The brain's resilience is rooted in its ability to separate physical hardware from computational software. When the tissue fails, the logic migrates. The adult brain cannot remain in a constant state of high plasticity. Rewiring tissue is an extremely metabolically expensive process. To trigger structural changes, the brain requires specific neuromodulators, such as spikes of epinephrine generated by intense focus and the acute stress of making errors during a task. Regular cardiovascular exercise also plays a critical role here, flooding the system with BDNF to chemically prime the brain for this structural remodeling. Even with these gates in place, the brain faces a synaptic paradox. If Hebbian learning strictly strengthens connections throughout the waking day, the total energy weight of the network would eventually saturate, leading to excitotoxic cell death. To survive its adaptability, homeostatic plasticity engages during non-REM sleep. The glymphatic system flushes accumulated metabolic waste, while the network undergoes synaptic downselection. The brain shrinks new connections, returning energy load to a sustainable baseline, while perfectly preserving relative strengths. Sleep operates as an architectural editor, locking in the signal of new learning by aggressively clearing the noise of the day. From the initial environmental stimulus all the way through to memory consolidation, human adaptability operates on a continuous, biologically rigorous loop. Functional plasticity provides the immediate agility required to shift heavy processing loads in response to novel challenges or sudden severe trauma. Structural plasticity follows up, physically cementing those temporary processing shifts into the permanent topography of the cortex. Yet this entire system would rapidly collapse without the rigorous homeostatic constraints of regressive pruning and sleep-driven downscaling. Adult neuroplasticity is a metabolically expensive operation that must be earned through intense focus, deliberate behavioral protocols, and optimized recovery. The human brain is an open, programmable architecture. Cognitive health and adaptability are not strictly dictated by age, but by how intentionally we engage the mechanisms of repair and growth.